Hello everyone, today I am going to talk about a few flow sheets as an example, and I will also tell you how to go about doing mass balance and heat balance for a unit operation. So, here this is an example of a flow sheet of alcohol manufacturing process. Okay. As you can see on the left hand side, you can see on the left hand side here the fermenter, that is the heart of the entire unit operation, where the fermentation takes place. Okay. Once the fermentation is over, the product comes down and there is a distillation which happens here. Okay. The distillation which happens here, and the product ethanol is collected here on the top, whereas the bottom contains all the solid material which is dried here, and then it goes as a dried product. So, there you have a rotary dryer, you have a spray dryer. So, as you can see, this particular downstream process for the fermentation of alcohol contains a distillation unit, and you have a filter unit, and you have dryers on the down, and here the product ethanol is collected here and further distilled to get very pure spirits here. So, this is a very simple downstream process. So, and you do not have many unit operations except for filters, dryers and distillation column. And as you can see, ethanol is a thermally stable compound. So, there is no problem, we can use the distillation column to remove the ethanol from the top. So, let us look at another downstream system. This is related to the acetic acid manufacturing. Acetic acid is manufactured both by the synthetic and the bacterial route, and the one which is manufactured by the bacterial route is what is used in the food as vinegar. Okay, 10% of the world's production is through this particular route. So, if you look at here, you have again on the left hand side the fermenter, then the filter. If you recall earlier, I said that filter is the first downstream process in after a fermenter or a bioreactor to remove the solids, the biomass, and then it goes into the extraction. You have a solvent and you have a train of extractor here, where the acetic acid is extracted with the solvent, and then again you have a distillation column where you get the pure acetic acid at the bottom and the solvent at the top, which is again recycled here for the solvent extraction process. So, here the downstream is a filter and a string of solvent extraction units. Let us look at penicillin. It is one of the important antibiotics, and as you can see, the downstreams are getting more complicated and complicated. So, you have the fermentation again here, and then removal of biomass using a filter, and then the supernatant, you are acidifying the supernatant, you are going into a centrifugal process here, and then neutralization. Here we have the extraction, crystallization, centrifuge, finally the product is dried, you get the penicillin salt. So, here as you can see, the product is a solid. So, obviously we need to go into centrifuge, and then we are collecting after drying, and then finally as a salt here. So, here the downstream contains quite a lot of filters, and then it also contains dryers as well actually. Now, let us go further down into complications. Now, we are looking at a monoclonal recombinant antibiotic. So, in that, as you can see, after the bioreactor we have the primary recovery, and then we have the purification, and then final filtration. So, initially again you have a centrifuge, and then we have another filter, two types of filter, as you can see, polishing filter as well as the ultrafilter. So, microfilter and followed by the ultrafiltration. So, microfiltration removes larger material, ultrafiltration removes smaller material. Then we go into now affinity chromatography, ion exchange chromatography, hydrophobic interaction chromatography. As you can see, a string of chromatographies where you are purifying your product, and then it goes into set of filtration, and finally you get your product here. So, here as you can see, you have filtrations, and then you have chromatographies, and then finally you go into again filtration to recover your product. Now, let us look at another product. It is called a recombinant human alpha 1 antitrypsin. So, after the fermentation, you can see there is a filtration. Now, here the product is intracellular. So, obviously we have to harvest the cells, collect the cells, that means harvest the cells, and then disrupt the cells using a bead mill, and then we are collecting the product by flocculation, centrifugation, and you are removing it using a salt precipitation, again centrifugation, then a set of chromatography here, and then final washing and polishing, and you get the pure recombinant alpha 1 antitrypsin. So, you see we have introduced a new downstream, especially a bead mill, because the product is intracellular. Now, let us look at a large number of amino acids. Amino acids are a multi-billion dollar business. They are used in animal feed additives like lysine, methionine, threonine. They are used in flavor enhancer like aspartame, serine, glutamate, monosodium glutamate. Then they are used in medical applications. So, large number of amino acids are manufactured using a biochemical route. Products like glutamic acid, lysine, methionine, they are the largest manufacturer. As you can see, millions of tons are produced actually. Okay, then you aspartic acid, alanine are also made through enzymatic synthesis. So, many amino acids are manufactured using biochemical process, and look at the raw material breakage cost. If you look at the manufacture of leucine and lysine and the breakdown of raw material cost, 67% is due to nutrient media, about 2% is because of the ion exchange chromatography, okay. Then 12% is because again ion exchange chromatography, and again you can see 6% is because of the ion exchange chromatography of various kinds. So, almost 20% of your raw material cost in the manufacture of leucine and lysine is because of the components used in the ion exchange chromatography. So, if I can reduce the amount of various raw materials used in the chromatography, I will be able to reduce the raw material cost. Okay. Now, let us look at another amino acid like leucine and lysine manufacture. Again, if you look at the equipment cost for manufacture of these two products, you can see only 33% of the cost is because of the reaction, whereas 28% is because of crystallization, 7% is because of an exchange chromatography, 12% is because of the filter, and 1% is because of the pump, and 19% is because of the storage. So, predominantly the equipment cost is because of the downstream. The reactors take only 33%, remaining 67% is because of the various downstream steps, filters, ion exchange columns, storage tanks, crystallizers, and so on. So, if I can optimize these, I will be able to reduce the total equipment cost. Let us look at again the aspartic acid and phenylalanine, but the raw material cost. So, if you look at the raw material cost, here 95% of the raw material cost is because of the nutrient, and only the remaining 5% is because of the various raw materials used in the chromatography. So, as you can see in the previous case as against here, the predominantly the nutrient media takes up most of the cost here. Let us look at the equipment breakdown cost for aspartic and phenylalanine. You can see reactors take only 30%, remaining is because of the downstream. You have the storage tanks, the crystallizers, the filters, heat exchangers, ion exchangers, pumps, and so on. So, almost 69% of your expense is because of those various equipments down of the reactor. So, we need to look at how to reduce those numbers and how to optimize their numbers, thereby I can achieve a reduction in the total equipment cost. Let us look at another important product, that is called human insulin. Okay, the human insulin is produced through various steps. The product is an intracellular, so that means I have to harvest the cells, I have to break the cells using some enzymatic approach, then I need to unfold and refold again, so that the insulin regains its activity. So, it is a very large number of steps involved in the manufacture of human insulin, and the 50% of the manufacturing cost is due to raw materials, 17% is because of equipment, and 13% because of consumables. Okay. As you can see, only 50% is due to raw materials, rest all is because of the equipment dependent and consumable dependent cost. So, I need to play around with that to reduce the total cost of manufacturing this human insulin. Again, if you look here, 62% of the equipment cost are because of the reactors, and 29% is because of the downstream. So, quite a considerable chunk of expense is because of the downstream steps involved in here. And this is a typical steps involved in the manufacture of human insulin. You are making your fermentation, the biomass, cell is harvested, and then the inclusion body contains your insulin, which has completely lost its activity. So, you need to unfold and again refold so that it regains its activity. These are the various steps. So, you have not only the fermentation taking place here, you have certain bio reactions taking place or biotransformation taking place, so that you get your human insulin in the active form. So, this is how the entire flow sheet looks like. So, you have the cell disruption, you have centrifugation, and then you have the bioreact, then you have the centrifugation again here, then you have the solubilization, you have the chromatography, two types of chromatography, again you are preparing the folded product, you have the chromatography here, polishing, and finally packaging. So, you end up with the product here. So, you can see two different biotransformations taking place, you are seeing a lot of chromatography taking place, and you are seeing centrifugation as well as the cell disruption taking place here actually. Okay. So, if you look at the breakdown of cost, 50% is raw material and rest all is related to the equipment dependent cost, which is 17%, consumable is 13%, water disposal, that means environmental remediation is 11%, labor is 7%. Okay. So, if you break down the distribution cost, reaction is 54%, primary recovery is 9%, final purification is 29%. So, the downstream primary as well as the final purification will contribute to almost 38% of the total cost of your product. Okay. So, what am I trying to tell? I am trying to tell you that downstream plays a very important role in the cost, both in the raw material cost as well as equipment dependent cost. So, one needs to really optimize, one needs to really minimize these costs, then only one can achieve a very competitive product with a good selling price, with the optimum selling price as well actually. So, let us look at some mass and heat balance. So, what is mass balance? So, if you have assume two streams entering a unit, this could be anything, it could be a filter, it could be a extractor, it could be an adsorber, it could be a chromatography, two streams, one stream coming in at a flow rate of F1 with a concentration C1 of a solute, another stream entering with a flow rate of F2 with a concentration at C2. Now, these two are leaving because of certain thermodynamic interaction, the solute gets moved from one stream to another. So, for example, if it is extraction, the solute will move from the fermentation broth right into the solvent phase. If it is an adsorption, it may move from the gaseous phase to the solid phase. If it is an absorption, it may move from the gaseous phase to the liquid phase, and so on actually. So, the two streams are leaving. So, you have the volumetric flow rate of F3, concentration of C3, volumetric flow rate of the fourth stream is F4, concentration of C4. So, if I want to do a mass balance of this unit, what do? I consider the inputs equal to outputs, that is what I do actually. So, if you are doing a volume balance, F1 and F2 are the streams entering, F3 and F4 are the streams leaving. Okay. So, F1 plus F2 equal to F3 plus F4. So, there is no accumulation inside the unit at steady state. Now, if we are doing a solute balance, what do? F1 into C1 plus F2 into C2 is equal to F3 into C3 plus F4 into C4. There is no accumulation, this is at steady state. So, if I have two solutes, then I will do species balance for solute 1, then I will do species balance for solute 2. So, these are very important set of equation, and we will be using these equations for solving mass balance in a adsorber, absorber, liquid extractor, chromatography. So, this is the volume balance, that is volumetric flow rate, this is the balance for the species or solute. So, if you have many species or solutes, you will have many equations of this form, and please note this is at steady state, that means there is no accumulation, we assume there is no reaction, there is no loss, there is no degradation. So, we need to consider all these. So, similarly we can do for heat balance is much more complicated because sometimes heat is generated when I add one liquid to another liquid. because of the thermodynamic mixing. Heat is lost to the surroundings, even though you may have a good jacket, you may have a good heat exchanging system, heat may be lost to the surroundings. So, we may have to consider that. So, here when you do a heat balance, we need to consider the specific heat of each of the streams. So, not only the flow rate and concentrations, we also have to consider the specific heat of the stream that is entering, specific heat of the stream that is entering 2, and specific heat of the streams that are leaving, streams 3 and stream 4. So, we need to consider all these, then only we can do a heat balance, and also we need to consider the heat loss and also heat generated. Okay. So, when we combine all of them, heat input equal to heat output, assuming there is no accumulation, that is what is called heat balance. Okay. That is very, very important. We do a mass balance first and then later on we do a heat balance later actually. So, heat inputs they are called sensible heats due to inlet fluid, heat added by heating medium. Sometimes we heat the contents of the unit operation because they may be very cold, sometimes we may have to even cool it because there could be a lot of heat generated. For example, in a bead mill or in a homogenizer, heat is generated, so we may have to cool them. Heat output will be heat removed by the coolant, so we may have a cooling medium, and then heat lost to the surroundings, and then heat removed due to the outflow of the liquid. So, we need to consider all these heats, and we say heat inputs equal to heat output, and that is what we do in a heat balance actually. So, we say heat input plus heat generated equal to heat output plus heat loss, that is what is heat balance is all about. So, if you have 2 streams, there will be 2 heat inputs, if you have 2 streams going out, that will be 2 outputs. If you have 10 streams input, then we will have to calculate the heat input for all the 10 streams, and if there are 10 streams going out, you have to consider heat output from all the 10 streams. Now, this sensible heat, how do you calculate? You calculate by taking volumetric flow rate into density into specific heat, that is called the Cp, into delta T. So, that is the temperature at which it is entering minus some baseline. We have to do a baseline, that means generally we take it as a 30 degrees or 20 degrees as our baseline, then it will be minus 20 degrees. So, we do that for both the streams that are entering as well as for the streams that are going out. So, the sensible heats are always calculated based on these baseline temperature. So, we need to subtract. So, if the temperature of the stream that is entering at 30, and if our baseline temperature is 20, we do 30 minus 20, and if the stream is leaving at 40 and if our baseline temperature is 20, then we do 40 minus 20. Okay. So, all the streams and the temperatures are subtracted by this baseline temperature. So, you need to remember that is very, very important. So, this is how heat balance is calculated. Heat balance is slightly more tricky when compared to mass balance, because in mass balance we are just calculating the volumetric flow rates, we are calculating the concentrations of the streams entering and equating it to the concentrations of stream that is going out. Okay. So, it is quite simple, whereas in heat balance we have to consider specific heat, we have to consider heat lost to the surroundings, we have to consider heat that is generated because of mixing of 2 liquids or 3 liquids, and then we also have to consider this baseline temperature which needs to be subtracted. Okay. So, you need to remember that. Why do you need to do this heat balance and mass balance? We need to do, as you go along, we will do some problems, you will come to know. For example, if I want to calculate how much solvent is required to remove a solute from the fermentation broth, if I want to calculate how much adsorber is required to adsorb certain amount of liquid or gas. So, if I want to calculate how much of stationary phase in a chromatography is required to remove certain amount of solute from the continuous phase. So, in order to do that sort of calculation, we need to do these mass balance. Okay. That is very, very important actually. Let us look at a simple problem which makes use of all these concepts, and which is very, very useful and very relevant to our particular downstream processing course actually. Okay, I want to tell you this problem. For example, streptomycin is produced in a fermentation, okay. Then it is filtered to remove the biomass, okay. Then after that the antibiotic which is in the filtrate is extracted using a low boiling solvent, okay, and then there is a distillation column where the low boiling solvent is removed at the top and the product is removed at the bottom. Okay. So, what are the downstreams? You have a downstream of a filtration to remove the biomass, then you have a extractor where a solvent picks up this product, and then the distillation column where your solvent is stripped at the top and the product is formed. Okay. So, these are the 3 unit operations. So, we need to look at the mass balance at each one of these 3 unit operations. Okay. Now, whenever you remove biomass in a filter, what happens is there will be some liquid in the biomass, because the biomass will not be 100% dry. So, there will be some liquid. So, when some liquid goes, product streptomycin will also go with that. So, you will lose some streptomycin. Okay. Now, when we go to an extractor, we are adding a solvent. Extraction is never 100%, even if it is 98%, what happens is some amount of streptomycin will go away with the parent liquid, it will not come into the solvent. Now, when we go to the distillation and when we are distilling, your solvent may come at the top, streptomycin will come at the bottom, but some streptomycin may also go away with the solvent. So, you will lose streptomycin at 3 different places. One is in the biomass filtration, and one is in the liquid extraction, and one in the distillation. So, you are going to lose this in each one of these stages. So, there will be total amount of streptomycin lost will be sum of all these 3. So, we need to do a mass balance for each one of these unit operations. That means, we do a mass balance for the filtration, we do a mass balance for the extraction, we do a mass balance for the distillation. Now, as you can see, this entire paragraph tells you what is happening whenever we do a filtration, whenever do an extraction, whenever do a distillation. So, that is what it is saying. The original broth contains 10 weight% of biomass and 20 weight% of streptomycin, rest all are mother liquor. That means, it will contain, suppose we take 100 grams, then 10 grams will be your biomass, 20 grams will be streptomycin, and remaining 70 grams will be liquid. Okay. Now, when you do a filtration, the solids will retain 5% of the solution, remember that. So, solids are always wet, in a filter we cannot completely dry. So, solids are always wet. So, when it retains 5, that 5% of solution will also contain some amount of antibiotic and some amount of the liquid, okay, like in the same ratio, the 10:70 ratio, sorry 20:70 ratio, because in the original broth you have 20 streptomycin and 70 mother liquor. So, you are going to lose some amount of streptomycin also in that liquid which is retained by the biomass. Okay. Now, you are adding 2 parts of solvent to recover the antibiotic by extraction. Okay. Now, the solvent extraction is not 100% efficient, it is only 97% efficient. That means, 3 weight% of the antibiotic that is entering the unit is left behind in the mother liquor. That means, it is able to take out only 97%, whatever is entering only 97%, rest of it is gone in the mother liquor. Now, when you are doing this distillation or stripping as it is called, now 2 weight% of the streptomycin that is entering is again taken away by the solvent in the distillation column. Okay. So, you have 3 steps, in each step you are losing some amount of streptomycin. So, I want to know how much of streptomycin is totally recovered from the original broth and how much is lost, that is what it is. And this is a very important problem, because we always come across such issues. We would like to recover as much product as possible, because the more you lose, more money you are losing, the more you recover, more money you are gaining, because you can sell them actually. Okay. If it is a very expensive product, each milligram, each gram will be very expensive. So, you cannot afford to lose any of them. So, let us look at the next slide where we are doing a mass balance for each one of the stages. So, we are looking at filter. As I said, I am having 20% of streptomycin. So, when I take 100 grams, I will call it 70 grams of liquid, 20 grams of streptomycin, 10 grams of biomass. So, the biomass captures 5 grams of solution. So, 10 grams of biomass is completely removed, but 5 grams of solution is lost. Now, in this 5 grams I am going to have 2 by 9 amount of streptomycin, because as you can see here it is the starting is 2, liquid is 7. So, 2 by 7 is the 2 by 9 is the amount of streptomycin here. So, how do I calculate 5 into 2 by 9 here, okay, that is 10 by 9. So, that amount of streptomycin is lost here. So, what is left behind in the mother liquor? I will have 20 minus, because 20 is the amount of streptomycin coming here. So, 20 minus 10 by 9 will be 18.9 grams. Now, I am adding solvent in the extractor, and it is I have told that 3% of streptomycin that is entering is lost in the extraction. Your extraction is only 97%. So, what do I do? 18.9 is what is entering, 3% is 0.03, that is 0.5675 grams is lost. That means remaining is 18.9 minus 0.567, that is 18.33 grams. Okay. So, now the amount of streptomycin, as you can see, has come down from 20 to 18.9 to 18.33. Now, let us go to the distillation column, the stripping column as it is called. Now, the problem says 2% that is entering is lost in the distillation. So, what is 2% of 18.33? 18.33 into 0.02, that is 0.367. So, what do? We say 18.33 minus 0.367, that is 17.97. So, you started with 20 grams of streptomycin in the original liquid with the biomass, okay. Then you have the filtration where we lost some streptomycin, we ended up with 18.9 grams. Then we went into an extraction, we lost some streptomycin, we ended up with 18.33 grams, and then we came down to the distillation where we lost some streptomycin, we came down to 17.97 grams. So, the overall efficiency will be 17.97 by 20 into 100. So, we end up with 89.8%. So, what does it mean? I have lost 10.2% of streptomycin in these 3 unit operations, that is quite a lot of streptomycin. So, what do? I need to improve on the efficiencies of each one of these. How can I do that? I can make my filtration more efficient, so that when I filter I will not allow too much liquid to stay with the biomass, that is the solid. I will try to make it dry as much as possible, so that all the liquid goes down into the filtrate. I can improve the efficiency of my downstream, so that instead of 97% I can make it 98 or 99. I can improve the efficiency of the distillation. So, instead of losing 2% I can make it less, maybe 1% or so. So, by doing each one of these I can improve the overall efficiency of the entire process. So, I may be able to move up to maybe 95-96% efficiency. I may not be able to achieve 100%, but I may be able to go much higher. So, this is a very simple problem, but it tells you lot of information, how to use mass balance, a very simple technique to understand which unit operation is very inefficient. So, I can focus on that particular unit operation and try to improve on it. I can also determine how much will be the overall efficiency of recovery, that is very, very important, because as a manufacturing scientist I would like to enhance my overall efficiency, reach as much as possible towards 100, because higher the efficiency, however higher will be the product recovery, and I will be able to achieve better profit into the entire process actually. So, this particular lecture which I have been talking about gave you a very brief rundown on various unit operations which are used in the manufacture of antibiotics and several other biochemical processes. People use sometimes filtration, sometimes chromatography, sometimes different types of membrane processes. Finally, if it is a solid you use a crystallizer, and then you use a dryer, and depending upon the purity requirement of the product you may have a series of chromatographic steps, as we saw in the manufacture of insulin and other antibiotics as well actually. So, now we will in the next course of lectures I will spend more time on various unit operations which are shown in these flow sheets to you. Thank you very much.